Hello everyone, welcome back to our channel. Today, we're diving into the fascinating world of biochemical tests, specifically focusing on the Benedict Test and the Starch Test. These tests are essential in the field of biochemistry, helping us identify the presence or absence of certain compounds in various substances. Let's start with the Benedict Test. Developed by Stanley Rossiter Benedict, this test is primarily used to detect the presence of reducing sugars such as glucose, fructose, and maltose. The test relies on the ability of reducing sugars to reduce copper, 2 ions to copper, I, ions when heated in an alkaline solution. This reduction causes a color change, indicating a positive result. To perform the Benedict test, first, prepare Benedict's reagent by mixing copper sulfate, sodium citrate, and sodium carbonate. Then, take the sample solution and add an equal volume of Benedict's reagent. Heat the mixture in a boiling water bath for a few minutes. If a color change occurs, ranging from blue, no reducing sugars present, to green, yellow, orange, or brick red, indicating increasing concentrations of reducing sugars, it's a positive result. Moving on to the starch test. Starch is a complex carbohydrate composed of glucose units and is a significant energy source in many diets. The starch test allows us to identify the presence of starch in a given substance. For the starch test, take the sample solution and add a few drops of iodine solution. If starch is present, a distinct color change occurs. The iodine interacts with the starch molecules, forming a dark blue or black coloration, indicating a positive result for the presence of starch. Now, you might be wondering, where are these tests used? Well, the Benedict test is commonly employed in food science to analyze sugar content in beverages like soft drinks, urine analysis in medical diagnostics, and various other industries to detect reducing sugars. On the other hand, the starch test finds applications in the food industry to detect starch in products like sauces or soups, in biology labs to identify the presence of starch in plant cells, and even in forensic investigations for analyzing stains on fabrics or surfaces. In conclusion, both the Benedict test and the starch test play crucial roles in biochemical analysis, allowing us to identify the presence or absence of specific compounds in various substances, aiding industries, research, and diagnostics. That's it for today's video. If you found this information helpful, don't forget to give us a thumbs up and subscribe for more informative content. Thank you for watching. Until next time, this the Curious Cosmos signing off.